Hi everybody, welcome to the statistics lectures for your dyspepsia module. My name's Tom Reeder. I'm going to teach you 3 stats lectures in this module, and I'm also going to do some workshops with you supporting the lectures actually in a different module in FAR 1004 a little bit later in the semester. So I'm actually an ecologist in the School of Life Sciences, not actually in the School of Pharmacy, but I do enjoy a bit of stats, which is why I'm teaching this part of the course. So feel free to drop me an email if you have a question about the course, and I'll do my best to answer. So the first lecture of this set of lectures is called What Are Statistics? And we're going to have a very gentle introduction to the basic idea of stats in pharmacy or in the life sciences more generally, since, like I say, I'm not actually a pharmacist, but what I tell you will be clearly relevant to your course. So the job for today, if I can get my slides moving, is to cover the following basic lecture outline in 3 parts. So first of all, we're going to look at the content of this section of the module, the learning objectives that we have, and the structure of the material and how it relates to the workshops I mentioned, as well as to reading, and a little bit about software that you'll need in the workshop. Then we're going to start on the content for the lectures, and the very first thing I want to do is have a look at data, the different types of data that you might encounter, and then what we might do with data, and in particular how we might use statistics to tell us something interesting about data. And there's sort of 2 main ways in which we can use statistics to look at data. One is for summarizing data, and the other is for testing ideas or hypotheses that we might have about those data. So in part 1 of this lecture, the first recording, we're going to look at the sort of basic information about the course structure, the learning objectives, and then we're going to have a quick look at different types of data that you might encounter. So just to put this in context then, the learning objectives for the 3 lectures as a whole are listed on the slide here. And in today's lecture, so that's the 3 20-minute chunks I'm going to do today, approximately 20 minutes, we're going to look at what kinds of data you might encounter. We're going to look at how to describe and summarize those data using both descriptive statistics and also graphs. And then we're going to look at what statistics are at a fundamental level, what they're for, not just describing data, but as you'll see, for testing hypotheses. And then when we go on into the following lecture, lecture 2, which is next week, we're going to look at in more detail at what this idea of a hypothesis is. What is a hypothesis? How do we test it? That's going to lead us to the idea of a p-value, which is a probability that we generate when we test a hypothesis. I'll explain what that is and how we make sense of it. Then we're going to go on to look at actually testing data sets with hypotheses in mind, testing hypotheses with data sets rather, and then we're going to actually look at some different types of tests that you might encounter. And these might be familiar to you, especially if you've done A-level stats, but even elsewhere you might have encountered, for example, the chi-squared test, a simple test we're going to look at in lecture 2, and the t-test, another simple test. We're going to look at how to do them, what they mean, what they're for. And then in the 3rd lecture, we're going to look at a different kind of statistical test, one called linear regression, which is designed for looking for trends rather than differences. The chi-squared test and the t-test help us look for differences in data sets. Regression helps us look for correlations. And the final topic in the 3rd lecture will be to look at confidence intervals that you see so often plotted on graphs or quoted when people are quoting statistics. We're going to look at what they mean and how to use them. Okay, so that's the sort of the objectives of this bit of the course. Hopefully nothing in there looked too terrifying. We're not going to do lots of maths. We're really focusing on using stats to interpret the kinds of data that get generated in studies of medicines and drugs and all of the things that are going to be of interest to you during your degree. The sorts of things we're going to look at in terms of testing will be the kinds of tests that you'll certainly be required to do a little bit in first year in the workshops, but then later on in your degree you might be using, for example, if you do a project where you've collected data and you want to test an idea about those data. So a reminder then, we're going to have 3 lectures, each of about an hour long. Each of them will be split up into bite-sized chunks for you. And then a little bit later on we're going to do a workshop which will be in a different module, like I said, and you'll be using Excel to put into practice some of the ideas that I've explained to you in the lectures. So although these are in different modules, they are closely related concepts or closely related sessions. and you'll need to sort of have some basic understanding of the lectures in order to be successful in the workshops. And at some point, hopefully later in semester, we will have a drop-in session to help support both the lectures and the workshops, probably more of a focus on the workshop really. People who've had a go at it and struggled will have a look at if you've got any problems, and also if you're busy revising for the exam, the lecture content in particular, we'll have a session where I can help you with that. The assessment, as I've already mentioned, as I just mentioned, is via the Rogo exam that you'll have at the end of semester in the summer exam period. So part of your exam will be questions about stats. And just to mention, as usual with this module and every other module, you'll have a chance at the end of the module through what we call Student Evaluation of Module, SEM, to comment on what you thought of the module. And by all means, in that feedback, do comment on the stats part of the course. Always good to hear what you think, what could be improved, what went well, etc., etc. So a little bit more sort of housekeeping that you need to know about. We will be not really following a particular textbook for these lectures, but you may want to look at a textbook to get some support in your understanding of the ideas that I talk about. So my suggestion would be a good starting place is Barnard et al., which is called Asking Questions in Biology. Yes, it's for biologists, or at least it was originally written for biologists by staff at Nottingham, actually, my colleagues. But it's a very general book, and although the examples tend to be biological, they'll be understandable to a pharmacy student, and they take you through the concepts and also how to deploy the tests that we talk about in Excel, although in their case they actually provide you with their own special Excel spreadsheet which does tests for you in a slightly intuitive way. So by all means have a look at that. Lots of copies in the library. There's an e-copy available, ebook copy available online too. But there are lots of other stats books, and if you prefer them, do have a look around what's in the library. There are quite a lot of different basic stats books aimed at scientists. There may be some aimed specifically at pharmacists. I couldn't find any really good ones, so that's why I chose to stick with the biology one. But really, it's a very commonly taught subject. So if you dip into any basic textbook on sort of basic first year undergraduate stats, you'll find answers to the questions that you might have from my lectures. Now you will need some software in order to do the workshops, but luckily it's pretty simple. You're just going to need Microsoft Excel. The only slight complexity is that you will need to install what we call the Analysis Tool Pack, which is an add-on to Excel. It's pretty straightforward to add it in. Everybody will have it. If you've got Excel installed on your computer, you can add the tool pack really easily. I'll explain how to do that later on. Okay, so embarking then on the content of these lectures, I think the first thing I really want to stress to you is that the reason why we need stats is because pharmacy, like every other science, really is a subject that's often about numbers, about data. And data are everywhere. Wherever you look in the sciences, you will find data. And we use the data to tell us important things about whether a drug works or what the side effects might be, for example. And so you can't hide from them. And they come in many different shapes and forms. And you'll notice, by the way, that I'm using data in the plural. There are data, not there is data. And you'll find that that's a distinction really between the sort of formal scientific literature where data is treated as a plural. I've used it as a singular there because I'm talking about the noun. But the word data in science tends to be a plural for data points, or strictly speaking, the singular is datum. So that's how I'll use it in these lectures. But you'll know if you are listening to kind of popular news articles or whatever about data in the public domain, often people use it as a singular. Don't get too confused by that. To be honest, it won't matter too much. But if you do write about data in your assessments or in exams, then I would suggest you try to write about the data in the plural. Okay, sorry, that's a real sort of nerdy aside. Let's get on with thinking about what kinds of data we see out there in the biological world, in the human body, for example. Well, this is a microscope image of some bacteria. Maybe there's some bacteria that have been isolated from a patient, grown up in the lab. We want to know what they are or something like that. And we've got a nice colour image here showing actually some molecular process tagged with different green and orange dyes. This might be reflecting levels of gene expression in these bacteria, for example. And from this image, we can actually extract all kinds of different types of data. So we could simply count how many bacteria are present. It might take you a while, but there are lots of data there, lots of bacteria there. We could look at each individual bacterium and ask something about that bacterium. We could measure how long the bacterium is, so that would be another type of data we could extract here. We could try to measure this expression, gene expression, if that's what this green and orange colouring means. Then we could try to measure the intensity of that colour in each bacterium, and that might tell us something about how strongly those bacteria are expressing a gene. It might be a gene, for example, for antibiotic resistance, which might be really important if you're thinking about treating a patient with a bacterial infection. At a more fundamental level, you might be interested in what species of bacteria these are. So there are lots of bacteria on screen, presumably all of one species, but you might need to know what species it is in order to know what antibiotic to use, for example. You might have lots of different samples like this from different patients, and you're looking for one particular bacterium that expresses a particular gene. That may be how you find it. Maybe you're looking for resistant MRSA or something like that, and you're using these coloured tags to identify the species of interest. So you might want to know for each sample, like the one on screen, is this person carrying the particular bacterium of interest or not? Is there an infection? And you could really drill down into the data displayed in this image, and actually each individual pixel on the screen could represent actually a series of numbers. For example, we could take the RGB colour spectrum for each pixel on the screen, and that might tell us something about the relative expression of the green and the orange dyes, which might tell us something about the biology of the bacteria. Okay, you don't need to worry about the microbiology of this example. The point is that from a single image here, we've got a whole bunch of different types of data, and hopefully as I was talking through, you were kind of conscious that these are not all the same kinds of data. There are different sorts of data available here, and so it helps really to think about data in different formal types. And there's a sort of major division between categorical and quantitative data that I want to deal with, and then within those types there are various sort of subtypes that we might think about. So categorical data are data where the values that you're recording indicate membership of a particular category. So for example, you might look at the genotype of a series of patients or of bacterial samples. You might have done a bit of genetics at A level, and remember that we might find if we had two alleles of a gene A, one was a dominant allele, capital A, and one was a recessive allele, little a, we could have different types of genotypes because everybody has two copies, one from each parent. So the ones listed on screen there will be the 4 different combinations that you could have. So you might identify the genotype of a bunch of patients at that particular locus, might tell you something about their health status or an inherited disease or something. And so each individual patient ends up with a category assigned to it, either homozygous recessive, homozygous dominant, or heterozygous. More fundamentally, especially if you're dealing with humans, you might find that some of them are dead and some of them are alive, or rather perhaps those that get a particular disease. I hate to use the COVID example, but let's say COVID. Some patients with COVID survive, most thankfully, but some patients will die. And so if you're interested in studying patterns of mortality caused by a disease, then that simple categorical data, dead or alive, yes or no if you like, is really important. So that's an example of a binary categorical variable. Binary because there are only 2 possibilities. So while there might be many different genotypes at a particular locus, for a patient you can either be dead or alive, you can't be anything else. So binary data are just a kind of special example of categorical data. But as you can see, there can be multiple possible categories or outcomes. You might, for example, be measuring patient outcomes on some different scale, not just simply alive or dead. Sometimes those categories can actually be placed in a particular order, and that gives rise to a special kind of categorical data called ordinal data. So ordinal data have some kind of intrinsic order. So anything that can be ranked can be treated as ordinal data. So very commonly, especially when dealing with patients, we might think of a Likert scale where we ask patients to rank how strongly they agree with a particular question, maybe about a symptom that they're suffering, from strongly agree to strongly disagree and variations in between. And those answers have a natural rank. If you're looking for how badly is somebody suffering from a particular symptom, if they say strongly disagree, then they're not suffering from the symptom. Strongly agree, they're suffering really badly, and in between you've got various shades of grey. So intrinsically those data could be ranked, and therefore they're ordinal. And so this is really quite common data that you get from patients in various types of questionnaires or surveys or medical records. Right, so those data are categorical. Each individual data point is a different category, and even though they can be sometimes put in a rank order, they aren't inherently quantitative. They don't quantify anything. But of course, in the life sciences in particular, there are loads of different types of things that we can measure on a quantitative scale, a truly numerical scale. And these can be broadly divided into two. We can have discrete data, where each data point occupies a particular integer value, so that is whole numbers, 0, 1, 2, 3. So the data are discrete because you can't have fractions or decimals in between the integers. Those are not possible. So for example, if I'm counting the number of bacterial colonies on a plate where I've plated out a patient's urine sample or something, and I've looked to see what grows, I could count the number of colonies and I'll end up with an integer value. There might be 50 colonies on a plate, or 100, or 3, but I can't have 0.6 colonies. It doesn't make any sense. So although I'm quantifying something with a number, I'm still dealing with a sort of a defined subset of possible numbers, the integers. These are discrete data. By contrast, we can measure things on a continuous scale. So we can use a quantitative numerical scale to measure something at potentially any number of possible values, an infinite number of possible values on a particular scale. So I might measure the concentration of a chemical or a drug. I might measure how long it takes for something to happen, how long it takes for a patient to have a positive response to a treatment or something like that. And time and concentration are examples of variables where basically we're in charge of how sensitive and precise a measurement we take, and we could choose to measure concentrations in millimolar or just in molar. We could measure time in seconds or in hours. So we're in charge of the units that we use, but there isn't inherently a set of discrete values that are possible and then a bunch of values that are not possible, which was the case when we were looking at discrete quantitative data like the number of colonies on a dish. Okay, so these major types of data help you start to make sense of the sort of vast array of data that you're going to encounter in the real world. So if we look back at our image of these bacteria, when we asked how many bacteria are there on the slide, well, we were asking a quantitative question. The data we're going to get, we're going to quantify on a numerical scale how many bacteria there were, but you can't have half a bacterium or 3.2 bacteria, well, not normally anyway, and hence these are discrete data. They come in integer form. Whereas when we measure how long a bacterium is, that's still quantitative. We're measuring it in numbers, but this time it's on a continuous scale. We can choose to measure the length of a bacterium in micrometers or in nanometers, different scales, potentially an infinite number of possible measurements given different levels of precision. So continuous data. The same is true of how strong the expression of this gene might be, so how brightly the colours that the bacteria are displaying. That could easily be measured on a quantitative continuous scale using a machine called a spectrophotometer, which measures the colour of light and the strength of that colour. By contrast, if we want to know what species of bacterium is present, then the name of the bacterial species, Clostridium difficile or something, is a category. So if we've got a whole bunch of patients or indeed a whole bunch of bacteria and they're perhaps different species, then each time we get a species name back, it's a categorical variable value that we're getting. Similarly, if we're looking at different patients, different samples, and we're looking for infections with a particular bacteria, then either you're infected or you're not. So that's a kind of binary variable, and it's categorical again. There's two categories, yes or no, infected or not. They're not numerical, they don't lie on a continuous scale. And I mentioned that the pixels themselves, you can actually extract the colour information, the data from each individual pixel here. And again, that would normally be on a continuous scale. Using a computer, you could quantify it approximately using the RGB colours that you can get back from a pixel. Okay, so that is the end of the first part of this lecture. I'm going to take a pause now, and then I'll come back and talk to you some more about how to summarise data.